I'm very pleased to introduce our first speaker, which is Dr. Mirik Briss, who is a movement disorders neurologist and neuroscientist who, for the past 3 years, has been working at Biogen as a medical director in early clinical development. He did residency and movement disorders fellowship at NYU, where he worked as an attending physician and served as principal investigator on several academic and industry-sponsored clinical trials. He also ran the NYU Neuroplasticity Laboratory and developed protocols for non-invasive neuromodulation techniques for Parkinson's disease. And in his industry role, he's been a clinical lead on several clinical development programs for Parkinson's, including the large multinational phase 2 SPARK trial, which you'll hear more about what that is. And that's a Parkinson's disease trial going on now. He also engaged in several initiatives aimed at developing digital endpoints for remote monitoring of Parkinson's disease using wearable devices. He's published over 40 papers and chapters, and also holds, in addition to an MD, a PhD degree. And he actually has both industry and academic credentials. He remains an adjunct associate professor of neurology at NYU. So thank you very much for speaking to us today.
Thank you so much, Dr. Simon, for this kind introduction. I also want to, tell you that how much I enjoy those presentations. When I was a full-time faculty at NYU, we used to do those make those events for our patients in New York, and it was an immense pleasure to interact with such a distinguished audience like you. So I want to thank you again for inviting me. It reminds me of my best days of my academic career, and it's, again, great pleasure to be here. Moving on now, as Dr. Simon has mentioned, today I present in a dual role, and that, of course, up front I want to disclose some conflict of interest. Of course, the one very obvious is I'm an employee of Biogen right now. The second, really important one that sets the stage for this presentation is that today we'll be talking about investigational compounds. We're not talking about therapeutics per se. None of these therapeutics or compounds is actually already approved. We're still in the phase of testing the safety, of course, testing the efficacy, or not even yet testing efficacy, and actually in some of those compounds are not ever, not even delivered for the first time ever to a human being. So please keep that in mind, because I want to make sure we don't overstate anything here. Those are all investigational compounds. Okay, so that sounded like a kind of a bitter start, but now let's go to more enthusiastic and actually give you a highlight of the presentation that I will be talking about today. So I want to start, as you noted in the title, two main proteins we'll be talking today about. I assume many of you, not all of you, have heard about at least one of them, namely the alpha-synuclein, abbreviated as ALPHA-SYN, as you can see here on that slide. And I'm going to tell you a little bit about the physiological role of this protein, but also, importantly, what's the link to Parkinson's disease. Second, we'll be talking about LRRK2 protein, and again, what does it do in normal, in a normal state in healthy people, and what's the link to Parkinson's disease. Then we're going to shift gears and talk about the investigational compounds, the compound called BIB54. We use code names in industry, which tells you again how far away from being being able to have it as a medication. But nevertheless, this one already has a name, and the name is just recently announced. It's called CINPANEMAB. Again, it's still in a trial. And of course, I'm going to mention some other monoclonal antibodies developed by other companies, of course, with very limited information that I have. We will be talking about something very, very new. This is for the first time ever disclosed actually today, that will be the compound targeting alpha-synuclein called ANTISENSE oligonucleotide. Bear with me, I'll try to do my best to explain the mechanism of action. It's a very complicated name. When you see a picture, it will be much easier to understand what it does. We call it a silencing approach. I'm going to try to explain this a little bit more. And finally, we're going to talk about silencing approach for LRRK2 protein. And again, talking about our effort in that space, but also, of course, to be fair, mentioning other companies, at least one company working in that space as well. Okay, so let's move on. So alpha-synuclein, this is a family of proteins. There are three of them, alpha, beta, and gamma from Greek letters. Today we'll be talking primarily about alpha-synuclein. And alpha-synuclein is a protein that the role of it is only partially known. We know that this protein participates in some neurotransmitter release. That means that is involved in how brain cells communicate with each other and how the chemicals are recycled when the message is sent from one cell to another. But there are also many other ways where alpha-synuclein is important. And for example, that includes some signaling between cells and not to, list all functions of alpha-synuclein. We know that synuclein, for example, is very abundant, in red blood cells. It's present in the gut. It's present in the pancreas. What does it do there? I don't know. I'm not sure we actually know, and this is a big question we're still asking. So, of course, that's important when we develop treatments that target this particular synuclein protein. Now, moving to what's the link to Parkinson's disease? Of course, first, it was discovered that mutation of this protein was found in several families who have Parkinson's disease. It's called autosomal dominant Parkinson. That means that only one parent needs to have the gene, and it's transferred to the offspring. And those, very often, if not always, develop Parkinson's disease. But importantly, what you see here on this picture is the picture of a Levy body. Levy body was a pathologist, actually a neurologist and pathologist, who described those clumps of proteins within cells, and the main constituent of this aggregate right here is actually alpha-synuclein. So you hear about Levy bodies, continuously in the United States. Sometimes, they pronounce it Lewy body. I've been told it's actually Levy body, that's actually, you see those clumps primarily consisting of alpha-synuclein. Now, the second protein is a little bit more complicated, and we know actually much less about it. The leucine-rich repeat kinase 2, abbreviated as LRRK2 protein. It's a very, very long and big protein. You have some schematic representation of this protein here. Again, this is beyond the scope of this lecture to go into all those domains, but all I want to mention is that this protein has many, many, many functions. It's encoded by the LRRK2 gene, and that's important because we're going to mention in a second that this is probably the most common reason for genetic form of Parkinson's disease. Interestingly, there is a Basque word called dardara, which means trembling, and actually in some publications, this is how this protein was called. I mean, I have never seen it myself, but nevertheless, interesting fact. Nobody uses this name anymore, but it's interesting that it's called named after trembling. Why? Because it was discovered in, again, many families who have this mutation of this protein, and not only the mutation itself, but also the levels of this protein in certain body fluids, activity of this protein in body fluids is significantly increased. What is even more interesting is that not only those patients who have a mutation of this protein, but also patients who have what we call idiopathic Parkinson's disease, non-genetic Parkinson's, the common Parkinson's disease, they also sometimes have high levels of this protein, and we'll be talking about this in a second. Okay, let's move on. So first I want to talk about immunotherapy, and we're gonna mention the alpha-synuclein again. So let me start with this. Why symptomatic therapy is not enough? We have multiple options. Levodopa is probably the best medication ever discovered. Nothing works as usually, at least early in the disease stage, as well as levodopa. There are other options listed here, but we know that levodopa and all those other medications, with time, they do not work as well as we want them to work. So that means the disease still progresses, and what we would really like to have as a treatment option is something that would actually modify the progression or slow down the progression. And I believe Dr. Simon will be talking about this in his talk a little bit later about progression. So when we talk about disease-modifying treatment, then immunotherapy as one option comes into mind, and I'm going to explain how this might potentially work, because again, it's a still hypothesis at this stage. But what disease-modifying option ideally in a best-case scenario would do, some benefits would include, for example, delaying the time when symptomatic medications are needed, lowering the requirements for levodopa symptomatic medications. If you lower the requirements for levodopa symptomatic, then you can think of all the complications from treatment of levodopa, maybe also at least delayed, perhaps if not limited. Of course, we talk about disease milestones. So milestones, when you start developing balance problems, when you start falling, when you start needing a wheelchair or a cane. Again, in the best case scenario, again, this is a dream, such immunotherapy or disease-modifying therapy, any other therapy that is changing the progression of the disease would perhaps help us with that. Okay, so moving on. Early in 2000, German pathologist Heiko Braak described certain pattern of spread of pathology in the brain, actually not only in the brain as we learned later, but initially described as in the brain, where pathology, namely composed of those clumps of protein called Lewy bodies, spread in a specific certain pattern. And how did he discover that? So he and his wife actually work in the laboratory, and they're doing autopsy. They were like analyzing the brains of Parkinson's patients with Parkinson's disease under microscope, and realized there is a certain pattern that, again, without going into too much detail, starts in the lower part of the brain, and then as time goes by, it progressed to higher up, eventually reaching the brain cortex, which is the surface of the brain. So that led to a subsequent theory that perhaps there is some, maybe like almost like a virus-like spread, when one cell would almost infect like the neighboring cell, transferring this, whatever that is. They call this prion-like hypothesis, virus-like hypothesis. I think I don't like any of those terms because, there's still a hypothesis, but nevertheless, there is some merit to this theory, and that's important that we think that this might be one of the mechanisms of Parkinson's disease. Now, so you may ask, okay, so where does it start? And again, there is some debate going on. Does it start really in the gut? How do we discover that? I'm not going to go into detail. It's a very, very long story, but nevertheless interesting, and I would encourage you to perhaps read something about it, because that story itself is fascinating. Perhaps the nose is the site of the origin, or maybe both. In some patients' nose, in some patient guts, we still don't know. So if that's the case, then perhaps when we have synuclein, as depicted here on this side, on this left side of this slide, which is not natively present in this kind of random coil structure, which is kind of, specific conformation has in healthy people. And if this synuclein starts to change its shape and starts to aggregate, when it starts to aggregate, it may progress to formation of something that's called fibrils, and eventually formation of, again, those Lewy bodies that I mentioned before. So if we zoom out, remember this brain from a previous slide where pathology would spread from one cell to the neighboring cell, and kind of in a contagious-like fashion spread through the certain brain centers. You can imagine that. How about if we take antibody, which is technically is a small protein, but binds specifically to one protein, in this case alpha-synuclein, and would perhaps capture this synuclein, which we believe, again, is responsible for this splat in the most likely non-monomeric form. Again, I'm not going to go into biochemistry here, but capture this, forms of synuclein that are spreading, and by doing so, ultimately, look for reduction of this pathology, reduction of this clumps of the protein, and perhaps slowing down the disease progression. Again, a hypothesis, but it might have some merit. So what we did, the phase 1 study of CINPANEMAB, which is, again, a monoclonal antibody targeting alpha-synuclein, was a randomized, double-blind, placebo-controlled, single ascending dose study. So what that means? That means that if you see this, schematic representation of all these cohorts, like typical for, phase 1 study, we just give one single dose. To patients, initially healthy patients in cohorts 1 through 6, but then in the last cohort, cohort 7, to patients with Parkinson's disease. So we just give one single dose to evaluate the safety and see how it was well or not well tolerated, and evaluated certain parameters, which of course I'm jumping ahead, but they gave us a green light to progress to subsequent stages of clinical development. Now, on my slides, I frequently refer to two things which I encourage you to do. One is that this is your friend. This is a government-funded website, clinicaltrials.gov. Whatever I present here is primarily coming from that website, and we also give the identifier, which is the study number. So I encourage you to go to this website in case you have questions and look for more information. There is no need to take notes. Majority of what I'm presenting today, you will find there. And of course, the publication is all the paper is published presenting those results as well. So you can also look this up. So now moving on, this is the schedule of assessments. It was a very heavy study. I'm extremely grateful to our patients who participated in this study. In the upper panel, you see the clinical examinations. EAC, there were so many of them. It was a 16-week study with multiple clinical exams, multiple blood tests, MRI, dopamine transporter scan, and importantly, lumbar puncture. So what we learned from this study that, before I actually go there, who we studied, particularly, we studied patients with Parkinson's disease 40 to 80 years old, diagnosed with Parkinson's disease and a HON-NER stage less than 2.5. So, that may sound like a simplification, but basically patients who do not have profound balance problems. The patients were treatment naive, so typically not taking any medications, or taking a stable low dose of selegiline, razagiline, or levodopa carbidopa. Again, there was a very low dose that they were allowed to take. We studied 18 subjects with early Parkinson's disease. They were randomized to one of those three arms of the study. One was, of course, placebo, as we always do in studies, and the other one was low dose of the antibody or high dose of the antibody. This study was done in 7 study sites in the United States. So what did we learn from this study? We learned that the most common side effect of this medication was headache. Of course, the question comes, if you have lumbar puncture, is this headache perhaps coming from the lumbar puncture? The real answer is we don't know that for sure. It might be, of course, the medication itself. And as a matter of fact, you seem that even in those patients who got placebo, then the headache was still present. So the majority of the adverse events were considered not related to the study treatment, in the opinion of the investigator or the clinician who ran the trial. We have no Parkinson's patients who experienced any serious adverse event who was receiving the medication with the BIP54 or CINPANEMAB, as it is now called. And headache was the only adverse event that occurred in more than one patient in any treatment group. We did have, however, one serious adverse event in Parkinson's patient that was an asthma exacerbation. However, this patient was in the placebo group. That means that this patient did not receive active medication. We did have also a serious adverse event in the very high dose of this antibody in a healthy in a healthy group, not in Parkinson's patient, but that goes beyond the scope of this presentation today. And again, the results are fully now published. The study is, of course, closed. All results are disclosed in the publication that you can see here before. That led us to a subsequent stage of development for this antibody that's called SPARK trial. Dr. Simon mentioned that before. This is the phase 2 study where the primary objective is still dose-related safety. So that means that even though many pharmaceutical companies in the phase 2 would already be looking perhaps for the efficacy, the primary objective is for us is safety. Of course, we're looking at efficacy, but the primary objective is safety. On the map here, you see the location of study centers, both within the United States and, and in Europe. You also have some list of secondary objectives and outcomes. So what we'll be looking at the brain scan, the dopamine scan, and we'll be actually quantifying the amount of signal in the brain using this specific radioactive tracer, and comparing how does it change for those patients who are treated versus those who received placebo. We will be looking again at the blood biomarkers and the CSF markers. So most of you, all of you probably are familiar with the UPDRS, Unified Parkinson's Disease Rating Scale. That was, of course, part of it. And we have something that's called quantitative motor assessment. I'm going to mention this in a second, using certain wearable sensors. This study is 357 patients. Again, I mentioned 9 countries, 75 sites. We study patients who are diagnosed within 3 years prior to screening, and patients who have not received Parkinson's disease medications. Now, this study is already fully recruited. This is the link to the website for this study, and this here on the right-hand side, you see the schematic representation of the study conduct. So you have right now we are within year 1 of this study. Subjects are randomized either to placebo arm or low dose, middle dose, or high dose. However, when the subjects complete year 1, they will be re-randomized, so no patients will be receiving placebo anymore. All patients will not know what's the exact dose they will receive. However, we'll make sure nobody is actually receiving placebo, and the study will be scheduled to continue for subsequent years. Now, I mentioned wearable technology. So, it's a buzzword, used very, very frequently. You all have heard about Fitbits, about Apple Watch, about, all those devices. Here, in this particular study, we use digital sensors to help to quantify some symptoms of the Parkinson's disease. So in this study, we actually use a device called APDM. This is a snapshot, a picture of this device. It looks like a watch. It has, electronic components, accelerometer, gyroscopes. Those of you who perhaps are part of the SPARK trial may know that there are the 5 sensors that we attach to, 1 to each wrist, 1 to each ankle, and 1 over lumbar spine, and the subject or patient is asked to perform a series of tasks. We collect the data in a very, through very sensitive, electronic components, and then analyze it offline with a quite sophisticated software. And of course, that leads us to quantification of certain parameters of the disease, like tremor, like postural stability, balance, turns, gait, and many, many, many others. Some of them actually we develop in-house at Biogen. Now, There are other immunotherapies in clinical development. Again, my knowledge is very limited. I encourage you to all those I know, which is publicly available. Those are the companies that work in that space. Roche obviously is the most advanced in that because they are already in the phase 2 clinical trial. The trial is called Pasadena trial, but there are many others, including Lundbeck, Takeda, and AstraZeneca, and AbbVie. Importantly, there's also another immunotherapy that's called active immunotherapy, developed by a company called AFIRIS, which works more like a vaccine. Now, what's the current status of this? I really don't know. I know that this company had completed some studies in Parkinson's disease, multiple system atrophy, but what are the plans? I can't really comment on that here. Again, I encourage you to visit clinicaltrials.gov, where you can find actually the information about those compounds. Now, there is something new, something else that we can work on, or another mechanism how we can target this synuclein. Now, remember how I mentioned to you that synuclein is important. So it's not necessarily a bad protein. We all have it for some reason. But sometimes, perhaps when you have too much of it, that protein tends to aggregate and form those Levy bodies and from the pathology. So, I mentioned, just before I talked to you about the immunotherapy, how you can capture some species of this protein as this being transferred from one cell to another. But how about if you take a different approach? If you don't just try to limit the spread, but try to lower the production. So actually your body and brain, most notably, produces less of this protein. And this is one example of this. So what we can do, we can use the technology called antisense oligonucleotide, that is abbreviated as ASO. So the way ASO works, and there are many examples of it, but in this particular case, all proteins produced in our body are encoded by DNA. DNA, however, need to be first transcribed to mRNA, which is another nucleic acid, and then only then mRNA can be translated into protein. So what does it mean? That means that the gene within the DNA tells the mRNA to provide a specific sequence of amino acids, which are the building blocks of any protein in the body, and only then the protein is being produced. However, what if you take a very short strain of artificially made, technically nucleic acid, which is the antisense drug mentioned here. Again, whether it's drug, it's an investigational agent at this time, and you bind to this mRNA and activate a series of enzymes that will actually start attacking or dismantling this complex. So effectively, this protein will not, this mRNA, this mRNA strain will not be able to result in effective production of protein. If that's the case, then of course you can imagine if producing less alpha-synuclein, then of course perhaps the aggregation or formation of Levy bodies might be limited. Again, that's the theory, of course, yet to be proven. Now, one important aspect is that the ASOs are delivered through a spinal tap, which is, of course, not easy. It would be much easier to deliver them either in the form of tablet in the best case, or IV, like intravenously, like that. But in this case, they do not easily pass through the blood-brain barrier. So what does it mean? If they were delivered into the bloodstream, they would never actually reach the brain, at least in the concentration we would want them to reach. So of course, then you need a spinal tap, which at the very minimum may cause some discomfort. Not to mention that you have to deliver those compounds repeatedly. However, I want to mention that there are many, many options how you can perhaps circumvent this step. And again, even though we're not ready as of today to have such option, this is an example of a device. It's not technically a delivery device. I just took it from a website of our colleagues at Medtronic who actually have a pump for delivery of agent for lower extremity spasticity when you have stroke. It's called baclofen. It's a pump. But just to show you an idea. So it's not exactly what I would envision or we can envision, but eventually we can perhaps circumvent the problem by, instead of you asking you or the patients coming for repeated spinal taps, we can have such a device, and then injection would be through a port, whether it's located under the belly, I mean under the skin on the belly or somewhere else. This is yet to be seen because, of course, such device has to be really customized. So having said that, this is the first disclosure. This was just published on clinicaltrials.gov literally maybe 2 or 3 days ago. This is the number. The study is called Horizon. This study will actually use the alpha-synuclein ASO, where again, we hypothesize that we aim at the reduction of alpha-synuclein production, and if that's the case, of course, the aggregation of alpha-synuclein may be lower and may perhaps slow down the disease progression. Now, as a first step, this study, this investigational compound will be tested in patients with multiple system atrophy, which is abbreviated as MSA. And again, this is a disease that I assume some of you at least heard. MS may have some similarities with Parkinson's disease. However, it's usually a worse disease. It's progressing faster, and our goal was to balance the benefit and risks, and therefore this will be a first study of the alpha-synuclein ASO, would be in multiple system atrophy and planned to start in 2020. Going on, now I want to briefly talk about LRRK2 protein and how we can perhaps reduce the production of that protein again with the ASO or antisense oligonucleotide. So that study, called a REASON study, is already ongoing. This study was started this summer. It's run by, again, Biogen, and the compound, again, like previous compounds, was delivered together with our partners at Ionis. The scientific rationale, and this is actually one example where, I apologize, there may be too many technical details on that slide. Being company representative, I'm only allowed to present it in a certain way. I cannot modify those. But what I can explain is that, again, within this approach, we are looking at, again, the reduction of levels of LRRK2 protein, which is implicated in the genetics form of Parkinson's disease. But also, there is some emerging evidence that also in the common or sporadic form of Parkinson's disease, this protein may play a significant role. But again, as always, in a first-in-human study, the primary objective will be to evaluate the safety and tolerability of this antisense oligonucleotide. Then will be injected through the spinal tap intrathecal injections to subjects with Parkinson's disease. As typical for first-in-human trials, we'll be looking for adverse events, serious adverse events, and many parameters that are mostly for our scientists at Biogen to plan a specific dose for subsequent studies and a specific interval, which is still may need to be perhaps more refined as we go, or maybe left unchanged. Now, that's again, I wasn't allowed to modify this slide. This are those the inclusion criteria, who is eligible to participate in the REASON trial. Again, those are literally copied and pasted from the clinicaltrials.gov website. Just speaking very broadly, we're studying patients with Parkinson's disease, both with and without the LRRK2 mutation. There are certain mutations that are eligible. Patients are screened for those mutations. And again, for those patients, they need to be diagnosed within the last 7 years without major fluctuations or dyskinesias. Again, we use the Hahn and Yahr scale, similarly to the study I was describing previously, and at this time the cutoff is 3. Again, there is no point of going through those exclusion criteria. I just was, have to present them in a certain way. I think that's important to mention that this trial is already ongoing, and we'll actually learn much more about the biology of this protein as this is given for the first time to humans, to patients with Parkinson's disease, and this will help us to optimize subsequent stages of clinical development. Now, to be fair, again, I would like to mention other approaches. Again, if you consider that the LRRK2 protein may have a role in Parkinson's disease, regardless whether this is the LRRK2 mutation or common Parkinson's disease, there is a small molecule. There's a small molecule means the medication that may be taken orally. Why? Because if you take a small molecule orally, that can easily go to the brain without any problems, as opposed to antisense oligonucleotide. And that compound, or actually compounds, I believe there are 2 of them, are developed by our colleagues at Denali Therapeutics. However, there are several others, at least molecules hypothesized to reduce the LRRK2 activity in a preclinical stage. That means they're not yet, at least to my knowledge, at the stage where they are considered to be given to humans. However, they may, I wouldn't be surprised if they enter the clinic, especially when there is more and more evidence that this approach may actually be beneficial to patients with Parkinson's disease. So this is my last slide. I think maybe I gave you too many details, but what I would like you to remember from this presentation today is that we have several novel approaches right now. It's a very unique time in Parkinson's disease research, in Parkinson's disease clinical trials, where we try to approach the problem from multiple angles. It's not only Biogen, there are many companies working in that space, and I think that all those approaches, as opposed to the typical symptomatic trials where they give you a transient benefit, symptomatic medications, they give you a transient benefit, they work very well. However, they just kind of cover up the symptoms, if you will. Those approaches will try to delay disease milestones. Now, the second thing I would like you to remember, that one challenge we are facing, again, all companies, everybody, all scientists and clinicians, and again, I believe Dr. Simon will probably talk about this as well, is how to measure this benefit. If you slow down the progression of the disease, for example, by, let's say, 20, 30, maybe 40%, how do you know how it's different from a noise? When you start taking medications that work very well, like levodopa, how do you know that the improvement you see is not perhaps coming from levodopa, even if you are asked to refrain from taking the medication when you come for clinical evaluation, right? We know that what we call the off evaluation, when you ask, the doctor may ask you to come in an off state, yes, it's usually worse than typical, but sometimes the effects of levodopa may linger for a while. So those are the challenges, and one way to approach those, we use technology, and I showed you an example of sensors, electronics, where they give you that more precise, kind of fine-tuned approach to look at this, not only in clinic, but perhaps also at home. So I could imagine there is a study run actually by Biogen as well called Watch PD study, where we use Apple Watch. This is an observational study. We study, up to, I believe, up to 100 patients with Parkinson's disease in over 1 year period of time. They're wearing Apple Watch. They still come for clinic for routine visits, and we'll try to learn what does the technology can tell us about how you function in a daily life. Something that we can't really see as clinicians when you come to clinic for a brief snapshot of disease. Another thing to remember is that, what we know that at least from the first human trials in the monoclonal antibodies targeting alpha-synuclein, they seem to be fairly safe. Those studies, again, were not very, very different in terms safety-wise that what we see in other monoclonal antibodies. This is a class of medication used very, widely in other diseases in other areas of other specialties in medicine. But perhaps the more robust strategy would be not only to capture those and to capture those bad proteins as they transfer from one cell to another, but to target the production. And show an example of antisense oligonucleotides, or ASOS. Even I cannot pronounce this, as you can even see, or ASOS. And you can actually see how that may lowering the production, hopefully, perhaps may lead to a clinical benefit, which is yet to be seen. And if needed, we can perhaps find ways to workarounds to make sure that you don't have to come to clinic for monthly or every few months injections of those compounds. So I will stop here. I hope I may spoke a little bit longer. Thank you very much.
Oh, sure. So we have time for a few questions, and we've asked people to write them down if you can. I guess there's some cards hanging around, and I'll ask them for you to answer. Actually, the first couple of questions both are the same, which is that you mentioned that alpha-synuclein and LRRK2 can be genetic forms of Parkinson's. Will these treatments targeting those proteins also work for people who don't have mutations?
Absolutely. So let me start with alpha-synuclein. The reason is we believe yes. It's a simpler answer because we know that Lewy bodies are always composed of alpha-synuclein, regardless whether you do have mutation in the synuclein gene or multiplication of the copy. So in other words, extra copy of synuclein or not. You always have those Lewy bodies, and you always have synuclein in there. So that I cannot imagine a reason why it shouldn't work, but of course that's a theory that yet has to be proven. For LRRK2, there is some evidence that patients with this, even without mutation of this protein, they have a higher level of either the protein or the activity of the protein is higher. So that actually led to believe that patients without mutation may also benefit from this treatment. Again, this is at this point something that has to be proven, but so the answer is yes. I guess there is a reason to believe that both genetic and non-genetic mutation forms may benefit.
Great. Thank you, and I can tell you we're getting a lot of good questions here, and I apologize if we won't be able to get to them all, but I'll be able to cover a few, or ask you to cover a few of them. Another interesting one is please compare the recently recalled drug for Alzheimer's to the presently discussed drug.
Right. So unfortunately, I can't comment much on Alzheimer. I'm a Parkinson's specialist. Having said that, we will learn more about this drug hopefully very, very soon. What I think is that what's important, the way those two drugs were discovered, so aducanumab and alpha-synuclein monoclonal antibody, CINPANEMAB, is kind of similar. They come from the same company and the same discovery platform. And, but that's about it. They target different proteins. The mechanism of the disease is different. So I would really discourage you from drawing too many analogies other than they probably come from the same source. So I'll be very, very, careful about drawing analogy between those two proteins.
Here's another quick question. How many years before the drugs you're talking about would potentially be available to patients if they work?
That's a very, very tough question. If you look at in the statistics or history, the typical drug development phase, clinical development phase for a compound is anywhere from 5 to 10 years. So even in the best case scenario of this drug, the SPARK trial, we're talking at least several additional years until we collect enough data and have enough patients observed for a sufficient amount of time that we can come to a conclusion.
So here's a question related to something you did touch on. ASO against alpha-synuclein, regarding that study, somebody points out that alpha-synuclein problems relate to misfolding, but ASOs would block all types of alpha-synuclein from being produced. Would that potentially lead to adverse effects from losing the normal function of alpha-synuclein?
Absolutely, and that's a very, very good question because everybody's asking this question. Whenever we submit the study for review, that's of course the question. Our answer is that we're not aiming at completely blocking the production of the synuclein. We're aiming at reduction of this production of synuclein. So we believe that in a certain parts of the brain, if you reduce by the production of synuclein by a certain amount, that probably would not exceed 50%, you may have some therapeutic benefit, but of course yet to be seen because that has not been studied yet. So again, the answer is we're not trying to block the protein from being made. We're trying to reduce the amounts of it.
Here's a question, hopefully I'm reading it correctly, is asking about imaging for alpha-synuclein. I think they're referring to imaging in the brain so you know whether or not you're having the effect you want.
Right, so this is actually a very sad and discouraging. We've been waiting for the imaging agent for synuclein for a long, long, long time. And just recently, the Michael J. Fox Therapeutic Conference, some results from were presented publicly, and the way I read it, at least personally, we're still far away from that. So there were some agents that perhaps may bind to synuclein and show you the signal. However, the signal is too noisy to be useful at this time.
And I think this will be the last question then. It says, during the trials that you've already conducted where, a subset of patients got the active dosing, are you able, what kind of data do you have regarding whether or not it did affect protein aggregation in the brain or affected progression in those patients?
Right, so we have some limited data yet. Again, some of this was described in the paper, so I encourage you to, for those who want to learn more details, to go to the paper. And by the way, the paper is available free of charge if you go to pubmed.gov. It's open access paper. Because if I would like to explain the details, it would take me a long time. But the bottom line is we do have some evidence from primarily from the blood, but also emerging some evidence from the cerebrospinal fluid, how those antibodies may work. But again, that is yet to be fully evaluated. This is a very long process to study this.
Okay, thank you very much.
Thank you.